So in this video, we're going to talk about cell respiration, and you've probably done a lot of talking about cell respiration, the whole process. Well, you can have like a table in front of you for this exam. They're not going to quiz you on the process, right? They want you to know for cell respiration the big ideas, right? The things that you don't just, you can't just have a table out in front of you in your notes, right? Things that you want to have to understand and be able to apply information from the cell respiration process. So I have these diagrams here, and it may look like I'm going to talk about the diagrams. I'm not. Okay? I want to talk about our three big ideas that in looking and analyzing at these models: the structure and function, the critical steps, and the regulation. Right? And so I just have the diagrams here so I can show a few key things about this. One key thing, let's go here. One key thing is this input of ATP. You have to have an input of ATP in order to get some out. So you end up netting, right, 2 ATP in glycolysis even though you get 4 out. So you end up harvesting, by the end of the process, about 36 ATP net. Numbers are going to be different. I know I originally learned 40. The numbers are generally between 32 and 42. But not super important. What's really important to know is things that you have to put in, right? You have to put in sugar, you have to put in energy, right? And you also have to put in oxygen. It's not here, but you have to put in oxygen, right? And that's going to be really important, right? Because if you want to regulate this process or figure out, how it works, you're going to want to maybe, the ATP will be hard to regulate because there's a lot of it in your body. But this process can get regulated by not having glucose or not having oxygen. The other thing is there are enzymes everywhere. Enzymes. Right here are enzymes. Big idea. Again, I can't promise anything about the AP exam because obviously I don't know what's on it, but I have a feeling that there's going to be something about some kind of enzyme regulation on this because it is so important in biology, right? Every process, as you've seen, can be regulated by some sort of enzyme, and you can relate it to a bunch of different topics. So I can't guarantee that that's going to be on the test, but it would make sense that it would be on the test. Enzymes are going to be another great way to regulate this process. Looking at this diagram, the main idea, the reason I put this here, and again, sometimes it shows 2 steps or 3 steps, sometimes 4 steps. Depends if you consider the link reaction or the chain reaction or the transportation stage, the transfer stage. If you consider that to be a part of the process, like an individual step, or if it's part of glycolysis. The big idea to know is where this is located, right? Glycolysis is outside the mitochondria. The rest is inside the mitochondria. And if we go down here to the electron transport chain, right? The idea behind the electron transport chain, the reason the structure of the mitochondria is so important, right, is you have this double membrane structure. You have this intermembrane space that allows for an electrochemical gradient to be established, right? So this is an incredibly important structure. And you have a lot of things going on in the membrane. And why is it going to be important that that you have these folds, right? The folds in the, I think they're called cristae, but in the mitochondria you have all these folds. Why are they important? It increases surface area to volume ratio, right? That's going to be important. You want to have large surface area, not a lot of volume. That lets you do a lot of things on the membrane, right? Because it lets you go through this process in the membrane a lot, because you have a lot of membrane, a lot of surface area to work with, without taking up too much space. Efficiency is the key. So that's another way. The reason it has those folds, it helps efficiency. That actually really increases the fitness of the organism by having a bunch of folds in there to have a really high surface area to volume ratio. The only reason I have this picture of the Krebs cycle is you might get this on the AP, right? They've shown the Krebs cycle or the Kelvin cycle in the next video, and ask you to analyze it. The important things to know are there are enzymes everywhere, right? Enzymes are proteins, right? There are proteins everywhere. And there are things you have to input into this cycle, right? You have to input acetyl coenzyme A, and you have to input, actually, you don't have to input oxygen here. The last big thing about regulating this process is this oxygen, right? These steps, everything after glycolysis, are called aerobic. I think it's aerobic. And these are oxygen dependent. Right? So if you don't have oxygen, these parts, everything after glycolysis, will not happen. And it will go into fermentation, which is going to be bad for the organism. Right? So O2 is going to be incredibly important in making sure this process happens, because O2 is going to be your final electron acceptor, right? It's your final electron acceptor. That's what pushes the electrons through this whole process. The electrons start here, and then as it goes into the mitochondria, it goes through a whole process that ends up with the electrons in O2, right? The electrons get to O2, and that forms H2O, right? So that's going to be what's really important, right, is you have oxygen at the end. That's your final electron acceptor. That's what powers this whole reaction, because you can't have the electrons flow through something without any place for them to go, right? And that's sort of the logic behind this. The reason oxygen is so important is if the electrons have nowhere to go, you end up having this process get held up, and your body resorts to fermentation, which is bad.